Okay, yeah, I will talk about our tool PanBARLEX, which is specially for barley, for the pangenome of barley. So it's not a generic tool. And to get this, I have a little problem with this bar here of Zoom. Okay, yeah. So the barley pangenome consists of 76 genotypes, and all of those genotypes are annotated with genes, which leads to 3.8, roughly 3.8 million genes. And for all these genes, we have five sequences: protein sequence, coding sequences, transcripts, etc. And this leads to a number of roughly 19 million gene sequences. And all these data is available on PanBARLEX, which is a gene-level comparative visualization and analysis tool for the Barley Pangenome version 2. And here you can see the start screen and the URL where you can access it. And the main goal of the creation of PanBARLEX was to democratize the access to the Barley Pangenome and to have also a one-stop shop to check allelic diversity, presence, absence variation, copy number variation, and gene expression data, and also have some useful tools for ad hoc analysis and customized data download. Um, so there are several entry points for researchers and breeders. So you can directly enter via a gene ID from the official annotation of the Barley pangenome. You can search via a full text search for functional annotations of the genes. You can enter your specific coding sequence or amino acid sequence to perform a blast search against the gene space of the Barley pangenome. Then we have also a known gene pages with 104 clone genes, where we have also connected the publications or the papers and the corresponding coding sequence clusters. And then we have also a gene ID leftover page, which I will show you later. And we have a protein embedding page, which I show you also later. This is what a gene details page looks like. So these pages are available for all 3.8 million genes. You can see the position on the chromosome. You can see the gene structure from the annotation. There's the functional annotation available, how many transcripts, etc. Also, there's a link in this row variants to our variant browser, which links the pangenome to the bigger diversity space of more than 1,000 genotypes that have been genotyped with whole genome sequencing. And also on this gene detailed page, you can see, sorry, you can see the sequences and the distinct features like exons. Um, and a big problem in the Barley community is the so-called evolving gene ID problem. So we have for the same gene now multiple different IDs from the different reference genome versions before the pangenome and within the pangenomes. And for example, all those IDs you can see on the top left are encoding the same gene, which is alpha amylase. And to overcome this problem for the researchers, breeders, and scientists, we have developed here a so-called gene ID liftover between all the different versions of the gene annotation. And this is what it looks like. So you can enter your gene ID here on top, which can be from one of the different gene annotations before the pangenomes, Morex version 1, 2, and 3, and the two Barley pangenome annotations. And then you can execute the search, and then you get the results. And you can directly see in which annotations your gene, your favorite gene, is also present. And then there are links to other systems like the JBrowse instances on grain genes and also PanBARLEX. Also here quality metrics are visualized for the alignment between both gene IDs. Now I will come to the so-called homology clustering. So to get meaningful clusters from these 3.8 million genes, we have introduced a workflow, which is displayed here. So everything starts with the 3.8 million pangenome coding sequences, where we have the gene details pages available. And then to these coding sequences, a clustering is applied using MM62 software, which is specific hyperparameters which have been aligned with prior knowledge. And this leads to a catalog of so-called coding sequence clusters. And also on PanBARLEX, there are distinct pages available to crawl, to search or to investigate these sequence clusters. Also, these sequence clusters are integrated with the so-called PAN transcriptome, which I will show you later. After the clustering, multiple sequence alignment have been applied to the derived clusters. And for the derived or for the multiple sequence alignments, a multiple sequence alignment viewer on PanBARLEX is available, where you can switch between visualization of coding sequences, of the aligned coding sequences, or the aligned protein sequences. And finally, from the multiple sequence alignments, um, phylogenetic trees have been calculated using FastTree and Clustral Omega. So you can also switch between these two versions of trees. So this is what the coding sequence clusters catalog site is looking like. So here you can see in each row a cluster, its functional annotations, and the number of transcripts, genes, and genotypes in the cluster. And also you can directly see the spatial distribution along the 7 chromosomes of Barley, so that you can directly see if it's maybe a gene or if it's a pan gene affected by translocations or um, other types of presence-absence variation, etc. Um, then if you click on the ID of the cluster, yeah, then you go to the detail page of this cluster, where you can see everything in much more detail. And you can also then activate multiple sequence alignment, phylogenetic trees, gene expression tab, and also download data. So the coding sequence clusters can be filtered by different aspects. For example, you can only show a single copy core genome genes. You can also show multi-copy core genome genes or shell genome genes or cloud genome genes. Then you can also filter for specific chromosomes. You can search for clusters which are annotated with a specific functional annotation, and so on. So the multiple sequence alignment viewer is here displayed in more detail. Here you can also see for each genotype if it's, or for the genes, if it's a gene from a cultivar, a landrace, or a wild type. And also you can switch between two multiple sequence alignment tools, which is MAFT and Clustal. And also you can switch here between coding sequences and protein sequences. The phylogenetic tree visualization widget allows you to display a tree based on protein sequences calculated by MAFT and FastTree. And here you can also switch to show a tree calculated on the basis of coding sequences and, for example, also on clustral omega. The Barley pan transcriptome has been integrated on the level of the coding sequence clusters. And this is because it gives you more overview instead of showing each expression level for each single gene. And here on the level of the coding sequence clusters, you have the greater overview, what is going on with the pan genes and the expressions. And here you can see for all genes where expression data was available and mappable. You can directly see the TPM values for each 3 replicates and the 5 different tissues, and also a mean TPM value in the bottom row across all genes and replicates. So this gives you um, mean level of expression for each tissue, which is more robust than the individual expression levels from the individual genes and replicates. Also available here is a visualization of the alignments of the assembled transcripts to the genome assemblies. And also you can here see classification codes derived from GFF compare and the original transcripts IDs. And on the same page, these transcript IDs are also linked to the PanBART website of James Hutton Institute, where you can get more information about the transcripts. Finally, on the coding cluster page, you can download data. Even you can download unaligned sequences as multi-faster files and also aligned sequences as multi-faster files for downstream analysis on your own. As I mentioned before, there's also a so-called protein embedding available as an entry point for users. And I here on the top show you the workflow to finally get these protein embeddings. So we start with the pangenome coding sequences and then perform the sequence clustering, which is the same sequence clustering as I showed before for the coding sequence clusters. But here only representative sequences from each cluster are taken and then fed into a neural network, a so-called protein language model. And then for each protein embedding vector is derived. And this embedding vectors are then visualized via dimensionality reduction on a two-dimensional representation, or simply called a scatter plot. And this two-dimensional representation can then be overlaid with additional metadata like gene expression or functional annotations, which is showed in the picture below. For example, on the left one, you can see resistance proteins, and followed on the next picture by transporter proteins and so on. So this tool is also highly interactive. You can enter your own functional annotation keywords and then click on highlight, and then in the scatter plot, these coding sequence clusters are then highlighted. And which gives you, for example, here that it shows you that these resistance clusters, coding sequence clusters, which are annotated with disease resistance, are locally nearby, which means that they are similar regarding structure, protein structure, and also potentially on a functional level. Here's another visualization type. You can overlay all the sequence clusters with TPM values for a specific tissue. Here I activated the embryonic tissue. And in yellow, these are the highly expressed genes, and bluish are the low expressed genes. And here you can investigate specific clusters and clusters on a superclusters, I would say, and also coding sequence clusters that are maybe not, that are dispersed more. And here you have also the ability to make a lasso selection around the scatter dots to get a list of functional descriptions of those selected coding sequence clusters. And also you get the list of the clusters with links to the details pages of the clusters. So this can then act as a tool for explorative data analysis of the whole gene space, which is far big currently. Okay, thank you very much. I'm looking forward to the question and answer section, and thank you very much for your attention.